So now we know what our taxonomy looks like and how we develop a taxonomy. The next question is, how do we organize our species, our organisms, into different taxa or groups? There are multiple sources of evidence that we can use. So taxa, or groups, can be based on several factors, some of which you guys probably remember that we studied in our evolution unit. So the first one I'm going to talk about is anatomy. Remember, that is structure. So we can organize our organisms based on the structures, their body structures. We can also use physiology. And where anatomy is about structure, physiology is about function. So we can categorize organisms based on functions that they have, such as circulatory systems, respiratory systems, and so on. We can also use embryology as our source of evidence to make groups. Taxa can also be figured out using the science of ontogeny. All of that means is development. We can look at stages beyond the embryo and see if two organisms develop in a similar manner. And last, what we're looking at to organize these taxa are evolutionary relationships. At our highest level of classification, we have domains. Again, there are 3 of them. We're going to make a chart that is going to describe how we can classify organisms into these domains. So the first thing I'm going to look at for each of these is whether multicellular organisms are included, or if it is just unicellular organisms. Then we're going to look at cell wall structure. Then we're going to look at whether a nucleus is present. And then we're going to look for membrane-bound organelles. And we're going to go through each of these for the 3 domains of life. Let's look at bacteria first. Bacteria cannot be multicellular, they're just going to be unicellular. If you remember from our virus and bacteria unit, it does have a cell wall. And I want to add a note here that it contains a special compound called peptidoglycan. Peptidoglycan. Peptidoglycan is chains of carbohydrates that are linked with very small peptides. Bacteria do not have a nucleus, and they do not have membrane-bound organelles. So now we can look at archaea. Just like bacteria, archaea are unicellular. They do have a cell wall, but it does not have peptidoglycan. They do not have a nucleus or any other membrane-bound organelles. So if we're trying to classify an organism and we're not sure if it's bacteria or archaea, we will have to look at the cell wall of that organism and see if it contains peptidoglycan. So the difference between the archaea and the bacteria are whether or not the cell walls have peptidoglycan. And the last of the 3 domains, Eukarya can be multicellular. Not all eukaryotes are multicellular, but they can be. Whether or not Eukarya has a cell wall varies. Our plant kingdom has cell walls, but other kingdoms like Animalia do not have cell walls. However, all of the Eukarya do have a nucleus, and membrane-bound organelles. So if we are looking at an organism and we see a nucleus with membrane-bound organelles, we know it has to be in this third domain. Some examples of our unicellular eukaryotes would be phytoplankton. So we can't say all of the organisms in the domain Eukarya are multicellular because some are unicellular. But in bacteria and Archaea, all of those organisms are going to be unicellular. Another way to differentiate bacteria and Archaea is that oftentimes Archaea are called extremophiles. The last part of that word is just like hydrophilic, water-loving. This time it's loving extreme environments. So you will often see Archaea at deep sea vents where it's super, super hot and super toxic, or you could see them in very, very salty environments. Just very extreme environments where you'd think nothing could possibly live there. Oftentimes, that's where you find the Archaea.